Uh, Happy day two. Here's to the beginning, huh? So, for this little video, we're going to be going over the first review question that you went over, the first practice for the AP exam. I'm going to read it. I'm going to go over all the answers that you were supposed to give, and you're going to get a sense of where you're supposed to be with this question. I will be grading it, and I'll be doing my best to figure out and give you like whether or not you'd be close to a 5, close to a 3, or close to a 1 on your response. I do not know exactly how everything would break down for this question. I do not know the specific conversion from correct to 5 or 4 or 3 or 2 or 1. So bear with me, but I'm going to go over all the answers. Here it is. First up, we start by reading the question. Okay. Scientists studying transcription in yeast, Saccharomyces cerevisiae, created an experimental strain that produced a modified RNA polymerase containing a single amino acid substitution. The scientists determined the maximum elongation rate during transcription with and without the modified RNA polymerase enzyme. The compound amanitin, which is commonly found in toxic mushrooms, is a specific RNA polymerase inhibitor. Amanitin binds to the RNA polymerase active sites and inhibits transcription. In a second experiment, the scientists treated the wild type and experimental strains of Saccharomyces cerevisiae with a 40 microgram per milliliter solution of amanitin and recorded the maximum elongation rate of the mRNA. Error bars represent plus or minus 2 standard deviations. Okay, and then you have 2 charts. One over here, wild strain versus experimental strain. And this is under natural conditions on the left, and then on the right, it was under the experimental conditions when they were in the solution of the amanitin. Whatever. Okay. Question A. First one. Describe the 3 structural components of RNA nucleotide monomer. Explain the role of RNA polymerase during transcription. Okay. B. Identify the dependent variable in these experiments. Identify control group missing from the second experiment. Justify the need for this control group in the second experiment. Okay. Describe the effect of amanitin on the maximum elongation rate of the wild type and modified RNA polymerases. Determine the ratio of the average maximum elongation rate of the modified RNA polymerase compared to the wild strand RNA polymerase in Figure 1. Okay, comparing data. Part D. State the null hypothesis for an experiment in Figure 1. Provide reasoning to justify the claim that the change in the amino acid sequence in the modified RNA polymerase affected the shape of the active site of the enzyme. Okay, that took over a minute just to read. Okay, remember, for the AP test, you're only going to have 25 minutes to go through all this. So be ready for it. Do not think you're going to have enough time to suddenly start looking stuff up. So, for part A, this is how you kind of break it out. So for that first question, how you're going to get graded is whether or not you included these things. So you needed 2 parts to answer that. Okay, you needed to just have a description of the RNA nucleotide and the 3 structural components. That means you have to bring up 5-carbon sugar for ribose. You have to bring up the phosphate group, and you have to bring up a nitrogen base. Now that could be adenine, cytosine, guanine, or uracil because it's RNA. Okay, so if you brought that up, all 3, you get 1 point. Okay, for the next part of Part A, you need to also bring up and explain the role of RNA polymerase. That might sound like this. RNA polymerase synthesizes a new RNA molecule based on the DNA template by matching the current DNA base with the proper RNA complement. Or, another example, RNA polymerase joins or bonds the newly paired RNA nucleotide and the growing RNA strand with a covalent bond. Unfortunately, you need to throw around some of that language in order to make sure you're getting credit on this. So if you're just saying RNA is made up of parts, or that RNA polymerase makes RNA, it's not going to be descriptive enough, and you won't get full credit for the AP test when you're answering like that. So make sure you're fully going into your science vocabulary. You already got it. You've been studying it. Okay? So That's part A. The second question, part B. Here we go. Again, this is now for 3 points. Okay, for 3 points. The response for the dependent variable, okay, is identified as the maximum elongation rate of the mRNA. All right, dependent variable is what you measure, maximum elongation rate. That makes sense. The next point you would need is an acceptable control group missing from the second experiment. You need to identify it, and here's some ideas that might work. The wild strain from the first experiment without amantine, right? Or the experimental strain from the first experiment without amantine. So you need to have a control group there as if you were running that lab. Lastly, the justification that the missing control is needed because the effect of amantine on the maximum elongation rate cannot be determined without comparison to the maximum elongation rate under the same conditions without amantine. Lots of words for saying that you need it there to make sure that you can compare, okay? So you needed all 3 of those. Explain like that in order to get credit for it, okay? Remember, as you're hearing this, think about how did you do when you did this as your homework? See where you are, okay? Part C, okay, 2 points again. You would need to first describe that amantine decreases the maximum elongation rate of the wild strain. It does not affect the rate of the experimental strain. All right, you need to describe that. And then the other part you would need is the ratio of the average maximum elongation rate for the modified RNA polymerases compared to the wild strain RNA polymerase is determined to be 1 to 6. Okay, something like that. You have to do anything in that range. Okay, you need to throw in some numbers there when it comes to some sort of ratio. All right, don't go crazy if you don't feel comfortable speaking like that or writing like that, but try your best to explain it numerically. All right, last part, part D. Again, 2 responses for this. Okay, you have to be able to you have to make a null hypothesis. Okay, you need to provide it. So a null hypothesis, an example would be the modified RNA polymerase would not affect the maximum elongation rate. Or another idea, any difference between the 2 elongation rates is due to chance. Null hypothesis means nothing happens. A null hypothesis means that you're saying that this doesn't matter. The variable didn't have any impact, like certain medicines that are being researched for, coronavirus. Last question, last point for this whole thing. A description that a change to the active site would explain the decrease in elongation rate is provided as reasoning to justify the claim. Okay, ladies and gentlemen, it's a whole bunch of points. So when it comes to that, I think that was 2, 3, 2, 2. You got nearly 10 different points you had to hit in that one question. Okay, did you see how it kind of goes over certain ideas where you had to explain the science? You had to kind of set up a science experiment. You had to then improve the science experiment. And then you had to know enough about what a null hypothesis is in order to answer it. Okay? So, hope you did well. I'll be grading them for this one. I will not be doing that for every single question coming into the future. Again, you're going to be doing some of this grading too. Okay? So, more work on the way.